So epigenetics is basically the study of changes in gene expression or phenotype without changes in the underlying DNA sequence or the genes. To talk about epigenetics, we talk about three things. The first thing we talk about is DNA methylation, then histone modification, and then non-coding RNA. In today's video, I'm only going to cover DNA methylation and a little bit of histone modification. So of course, to understand epigenetics, we must understand the epigenome. So the yellow strand here is the DNA, and when the DNA wraps around twice the histone proteins, we say that is chromatin. Okay? Now, DNA is negatively charged and histone proteins are positively charged, and that is why they can come together like this. Then we also see here chemical tags. And whenever you have chemical tags like this, we talk about the epigenome. And these chemical tags, such as a methyl group, can inhibit or can, for example, inhibit gene expression. So what happens is, for example, with the methyl group, is that the DNA gets wrapped around the histone protein more tightly. So when the RNA polymerase II wants to make transcription and read it, it cannot do that. So basically, you say that the gene is there, but it's silenced. Okay. However, on the opposite hand, you can boost transcription by unwinding the DNA. And one thing to understand is that all 200 cells in the body have the same genome, but it's due to gene expression that they're not all the same. Okay. Also, epigenetics is the reason why twins can have the same genes but be completely different because some genes are turned on and some genes are turned off, which of course varies from the first one to the second person. Then lastly, I want to talk about the fact that the epigenome is in a lifelong interaction with genes and environment. So these chemical tags can turn on and off genes, but these can be influenced by the environment, such as the diet, chemical exposure, medication, and sleep, and so on and so forth. So these chemical tags are influenced by the environment. So let's talk about 5-MC or methylation. So if you have cytosine and you add it you add a methyl group to it here, you get methylated cytosine or 5-methyl cytosine. And as I talked about, this makes the gene silent because the DNA wraps around more tightly around the histones, so the RNA polymerase II cannot read it. Therefore, some genes get silenced. Now, this process typically occurs in CGD nucleotides or cytosine guanine dinucleotides. And if you have a cluster of these, you call CPG islands. Okay. Now, CPG islands are typically placed in promoter regions, mostly. And this is all said because of the fact that 5-MC occurs not in random places, but mostly in CPG islands and promoter regions. Okay? It can also, of course, happen in other places. So this is CPG islands. Now you might ask, well, how do we add a methyl group to a cytosine? How does it occur? Well, it is catalyzed by enzymes. These enzymes are called DNA methyltransferases, or DMTs in short. Okay? We say DMT1 and we say DMT3A and DMT3B. So these are the main ones we're talking about. The DMT1 stands for DNA methyltransferase 1, and this plays a key role in maintaining DNA methylation pattern during cell divisions. This is responsible for ensuring that the DNA methylation status present on the parental DNA double strand is maintained in the newly formed daughter cells after cell division. Then these two play roles in de novo DNA methylation. So this refers to the process of adding methyl groups to a DNA where there was no methylation previously at the particular site. So in other words, it is a methylation that occur at new locations of the DNA where no methyl groups were present before. So here you have de novo methylation with these two enzymes, and here you have maintenance methylation with this enzyme. Then to repeat it again, you have cytosine, With these two enzymes, you make a new methylation, and with this enzyme, you maintain established methylation. Then you can also go in the other direction from 5-methylcytosine to cytosine. To do it passively, you can do it like this. You can also do it by TETs. TET, I'm going to talk about a little bit, but just to understand what demethylation here is when you're methylated and then you're demethylated, so you're unwinding it again. You're removing the methyl, so the genes are not silenced anymore. So then the RNA polymerase 2 can read it and transcribe it as normal. Okay. So TETs are called 10-11 translocation. These are 10-11 translocation enzymes. And these are a group of proteins that play a crucial role in a demethylation by catalyzing the oxidative removal of the methyl group from 5-methylcytosine. So this is one of the major epigenetic modifications in DNA. And one thing to understand here is that DNMT and TETs are in a constant battle with each other. Okay? Or typically they're balanced, but sometimes they can come in a battle with each other. So when you have hypermethylation, you have more methylation than demethylation, and this could indicate cancer cells. If you have more demethylation than methylation, you call that hypomethylation. Now, hypomethylation, it also, which is a reduction in DNA methylation, is also common in cancer cells. So you got to be aware of these things. Then we also talk about X-inactivation for DNA methylation. And this is when an egg cell is fertilized by sperm cells, so a zygote is formed, which is the first cell of the new individual, and this zygote undergoes some changes or cell divisions, which then results in the formation of this multicellular structure, which is known as the embryo. And here we see the embryo. We have the paternal X chromosome and the maternal X chromosome, and some random one of these gets inactivated. This is also why it's called dosage compensation for women, right? And then we get the Bohr body here. What's important to understand here is that under X inactivating, there can become there is an increased amount of methylation on the DNA in this inactive part. So one of the reasons why X inactivation happens to the X inactivated parts, so for example here the paternal X chromosome, is due to methylation. And lastly, let's talk about histone modifications. We have 12 modifications, 30 variants. I'm only going to talk about histone acetylations. Now, histone acetylation is basically where you're adding an acetyl group is added to lysine on a histone, okay? And this allows for relaxation of the chromatin and transcription can then occur. Then, of course, you also have deacetylation, removes the acetyl group and makes transcription harder. And then we have these two enzymes. We have HATs. So HATs is basically histone acetyltransferases, and these add acetyl groups to specific lysine residues on histone tails, leading to histone acetylation. Then on the other hand, we have HDACCs. These remove acetyl groups from histone tails, resulting in histone deacetylation, which typically leads to a more condensed form of chromatin. So this inactive chromatin, okay, or heterochromatin, where this one leads to euchromatin, typically.